Hi everyone, thank you for joining us for this webinar. Before we begin, I'd like to let you know that any questions you may have during the webinar can be typed into the chat box. And in order to get the best audio quality, please lower the resolutions on your screens. And with that, my name is Matthew Geniza, and I am a graduate student in Dr. Pankaj Jaiswal's lab. Today I will be walking you through an installation and operation of the Variant Effect Predictor tool using the command line and the Standalone Perl script. Now during today's webinar we will cover the featured data set, which are SNPs that come from wheat. We will learn how to download and install the Variant Effect Predictor. We will look at what type of input data that you will need to run the Variant Effect Predictor. And we will also learn how to run the Variant Effect Predictor on the command line. We will touch on caches and databases that will be required to run the Variant Effect Predictor locally. And we'll discuss what type of output data you get from running the Variant Effect Predictor. And finally, we'll talk about some downstream analyses that you can do with the output data. Now, I'd just like to remind you that Pankaj Jaiswal did a webinar back in July of Arabidopsis resources in Gramene, looking at SNP data and using the website version of the Variant Effect Predictor. Now, this version only accepts up to 750 variants. The software that we will be using today allows you to have much more data. And just a reminder, these webinars and video tutorials can be obtained by going to the Gramene website and clicking on the video tutorial link. Now I'd like to briefly touch on wheat. Triticum aestivum is the allohexaploid form, or commonly known as bread wheat. The A genome donor is Triticum aurar2, and it is diploid. Now the variety of wheat that we will be using in our analyses today is Triticum monococcum. It is also a diploid, and it is a part of the A genome family. Now these diploids can be useful in finding traits regarding salt, heat, and drought tolerance, as well as disease resistance. So they can contain many useful traits for breeding purposes. Now the sequence reads that we are using today come from a publication done by Sam Fox in 2014. In this study, we used Triticum monococcum subspecies DV92, which is a cultivated spring wheat, and G3116, a wild winter wheat. The sequencing was done on an Illumina HiSeq 2000, and approximately 37 gigabase pairs were generated per variety. Now, before we begin, I'd like to make you all aware that in order to run the Variant Effect Predictor, the Perl script standalone version, users will need to have some command line knowledge, whether that's Unix or Linux, and the user will also need to have provide their own SNP data set. Our SNPs were generated by aligning the sequence reads to the A chromosomes of the Triticum aestivum genome, which was obtained through Gramene. SNPs were called using the software VARScan to generate SNPs and indels, and we put those into VCF format, which can be seen in the picture below. Now, the Variant Effect Predictor also accepts pileup files and HGVS notations. So VCF isn't the only format that we use, but for this tutorial, that's what we'll be using. I'd also like to show you the Variant Effect Predictor homepage. This is where we can find all the downloads to all the software that we will be using. Now, in order to install the variant effect predictor, we will need to download the software from Ensemble, and this can be done by using the command wget on the command line. Once you enter this command, you should obtain output similar shown on the slide. And in the directory, if you do a quick ls, it will show you that the software has been downloaded. Next, we will need to unpack the variant effect predictor software, and that can be done by using the unzip command on the command line. Once you enter that command, you should obtain output that looks similar to this. And when you do an ls in that directory, you will see that the Ensemble Tools folder will be present. Next, we need to install the variant effect predictor on your local machine. First, we need to change directory to the variant effect predictor directory, which is within the Ensemble Tools file. These are the files that are located within the variant effect predictor. Using Perl, we will call the install.perl script and install the variant effect predictor. You will obtain output that is similar to the one shown on your screen. Finally, we want to test our installation of the variant effect predictor. This can be done by using Perl and calling the variant effect predictor Perl script. The -i notes an example VCF that is included in your installation package, and the cache option shows which database that you will be using. When you enter this command into the command line, you should see output similar to the one shown on the slide. A successful installation will end with finish at the very bottom of your standard output. Now, to run the VEP locally on your computer, we will need to make a few additional steps. First, we will want to download the species-specific variant effect predictor database from the Gramene genome homepage. This can be found on your respective genome's homepage, and there is a variation box that contains that information. If you click on the VEP link, it will lead you to the FTP site for those genomes, which can be shown here. For our tutorial, we'll want to download the Triticum aestivum variant effect predictor database, which can be seen here. If we right-click on that link, we want to copy the link address and store that for the next step. Once we have the link copied, we will want to unpack the variant effect predictor database. Now, I'd like to note that the variant effect predictor database will read from your home directory from a hidden file called .vep. This is where the species-specific database will need to be installed. Again, we will use the command wget followed by the copied link to the Triticum aestivum database. And once that file is in our .vep directory, we will need to unpack it using the command tar, followed by the options xvf. This should result in output similar to that is shown on your screen. Now, before we run the VEP on our SNP data, we need to make sure that the following have been done. First, the installation of the VEP must be successful. That can be done by testing the installation. Second, the local cache must be installed in your home directory within the hidden file .vep. Third, your variant file must be accessible and in your working directory. Now, to run the VEP locally on our wheat SNP data, we will use Perl to call the variant effect predictor script. Now, our options that we will need are offline, which enables offline mode, the directory of our cache, which can be seen here. The letters in red are my own personal directory, so that's just an example. Your home directory may differ. However, you will always refer to the .vep file for the cache. The species that we are running our SNP data against is Triticum aestivum, and the cache version that we are using is version 29. Our input file, which can be seen in red letters, must be in our working directory. This input file is supplied by the user. And our output file name will be generated once the analysis is run. So when we run this command on the command line, we should generate output that looks similar to this. Output will show how many variants are detected on each chromosome, as well as notify you when the output is finished running. Now, the variant effect predictor generates two output files. The first is a tab delimited file that has detailed information on the variant effect predictor for all of your SNP data. The second output generated is a web page summarizing the statistics for types of consequences, coding consequences, and distributions of variants by chromosome. Now, this is a snapshot of what the tab delimited form of your variant effect predictor output will look like. In the first column here is the data uploaded. The second column contains the SNP location. The third column contains the SNP allele. The fourth column contains the gene that the SNP is located in. And the fifth column contains the transcript that the SNP is located in. The seventh column here shows the predicted consequence of the SNP. Now, there are more details within this variant effect predictor file. Some that show coding consequences, as well as if your SNP matched a pre-existing SNP within the variant effect predictor database for that species. Now I will walk you through a little bit of what the web page summary will look like from your variant effect predictor output. On the top of the page, we can see some run statistics of the variant effect predictor, as well as some general statistics regarding how many lines of input were read in, as well as how many variants were processed. The web page provides summary on the types of variants that were generated. Here in our data, we have SNPs as well as insertions and deletions. We can also see the composition of consequences in our data, whether they are splice donor variants, missense variants, or intron variants. We can also see the composition of coding consequences in our data. Some range from stop gain to missense variants to synonymous variants. And finally, the web page also summarizes the distribution of variants by chromosome. Now I'm going to talk a little bit of how the variant effect predictor output can be used in your downstream analyses. For this tutorial, we will focus on a set of genes that have a certain predicted consequence of stopped gain. We will want to use the tab delimited file to look for all of the genes that may cause or that may have a SNP that causes a stopped gain consequence. This can be done by using the command grep and searching for the pattern stopped gain. Once you enter that command, you should get output similar to the one seen on your screen. Since we just want the list of genes, we can simply use the text editor command awk and print out the fourth column, which contains the genes that may have a SNP that encode for a stopped gain consequence. Entering this command will result in the following output seen on your screen. To find out more about the genes that have predicted stop gain consequences, we can use Biomart on the Gramene website. We can copy our list of genes and enter them in the gene stable ID query box. Next, we can select certain gene attributes, such as gene name, gene description, where the gene is located, or gene ontology annotations. We can also find information on these genes regarding protein domains using a variety of software and databases. Once we have all of our attributes selected, we can output this table, which can be used in further downstream analyses. This is very helpful in looking at what functions may be interrupted or affected when a stop codon is introduced, caused by a SNP. I want to thank you for joining us today, and just to remind you that this webinar, as well as previous webinars, can be found by clicking on the video tutorial link on the Gramene homepage. We also welcome any questions, comments, or feedback, which can also be found by clicking on the contact link on the Gramene webpage. Thank you very much for joining us, and we hope you have a great day.